Hello, two tenors! We've talked a lot about the benefits of active learning in this course, but you'll see with membrane protein transporters, there are pros and cons to both active and passive transport. We're going to look at the function of this large class of membrane proteins, and these can help regulate what comes in and out of the cell. This really is ubiquitous to all life, and we'll talk about the differences between active and passive transport, as well as primary and secondary active transport. We'll look at some great examples at how figuring out the structure of a protein can help you understand its function. Now, I've included this reference to Lord of the Rings not to refer to your performance in the course. We really do want you to succeed, but rather it's referring to that barrier that membranes set up with regards to allowing certain molecules to pass from one compartment to another. These are the supplemental readings for this lecture. Remember that membranes are lipid bilayers that set up this impermeable barrier that can only allow for very small, uncharged, or even hydrophobic molecules to pass. So you might sometimes hear the word lipophilic, which just means lipid-loving or hydrophobic. And if molecules really are too lipophilic, they might actually get stuck in the membrane and not be able to cross. Passive diffusion doesn't actually involve any membrane proteins or transporter systems. It's simply concentration dependent, where we have things going from a high concentration to a low concentration. Okay, so an example might be the waste product carbon dioxide that can diffuse out of cells where it moves down its concentration gradient. So in this case, we'd be going in the opposite direction. And we do know that this process is not saturable. So as you increase the concentration, there will be an increase of the amount going across the membrane, right? There really isn't anything limiting its movement across that lipid bilayer. Now, transport is really important for getting what the cell needs in terms of nutrients like amino acids or sugars or nucleic acids. That's just what's being shown down here at the bottom, as well as removing waste products, as I just mentioned. And it's really going to be these integral membrane proteins that are going to help move these molecules in and out of a cell. And we would consider this facilitated diffusion because these proteins are going to help or facilitate the movement of the molecules across the bilayer. Now, sometimes they might need a little bit of help in order to undergo these conformational changes that are needed, and that's going to allow the molecule to pass from one side of the membrane to the other. If we think about some of the ions or molecules that we talk about in biochemistry, here's an example of things that fly right through, like the gases. While water and urea are right on the edge, they can diffuse across, but not very well. So there are water transporters that can help regulate cell volume. These molecules have to cross the hydrophilic and hydrophobic portions. So when you start to build up to a larger polar structure or a charged ion, they can't get across. Not on here are the proteins, but you should now appreciate just how large they are since they are made up of many amino acids, and they can also be charged. So when you're looking in a cell, proteins really are confined to their organelle or their cellular location. If we left everything to passive diffusion, it would take forever. We need things to move quickly across the membrane for a cell to carry out its function. This is an example of a pore or channel that can mediate transport by facilitated diffusion. Because passive diffusion is quite slow, molecules need to pass first through the hydrophilic head groups and then through the hydrophobic regions. Membrane proteins help by creating a central passage that allows molecules and ions of the appropriate size, charge, and geometry to traverse the membrane in either direction. It's the amino acids that line this channel that will set up an affinity for specific molecules. For example, allowing sodium in but not potassium. You rarely have a situation where pretty much anything is going to get in. The cell just wouldn't like that. So integral membrane proteins are pretty selective for what they move into the cell by facilitated diffusion. Just like what we talked about for passive diffusion, this type of transport does allow things to go downhill, so from a high concentration to a low concentration, or in the thermodynamically favorable direction. The difference between facilitated and passive diffusion is that because we are dependent on a membrane protein to move things into the cell, you can hit a speed limit where transport is saturable. This is just shown here graphically, where along the x-axis is the concentration of solute, or the molecules that are moving across the membrane, plotted as a function of the rate of transport. That's what's on the y-axis. And you can see that as we increase the concentration of solute, for passive diffusion, we see the rate increasing, but it is quite slow. It's not until you have integral membrane proteins that are going to help speed up this process. Okay, and we can see that at very high concentrations, there's actually going to be a limit to how fast you can transport them, okay, or a plateau in the graph. So we do see this hyperbolic curve where you get to a point where if you add in even more of those molecules, the transport rate is actually going to remain the same. And we're going to see this is similar to what you'll see for a simple enzyme catalyzed reaction. Now, if you think about what's going on, there's going to be interactions occurring between the molecule and the protein. Okay, so if all of these binding spots are filled up, the protein can't function any faster. It has to wait until that molecule is left before it can interact with another one. Now, a good analogy would maybe be when you're trying to enter a TTC station, you know, not a really big major problem when it isn't rush hour, but if everyone's leaving class to head home at the same time and there's hundreds of people trying to get into St. George Station, those gates are going to be full and the lineup's going to back up out the door. Okay, so one way a cell can actually compensate is to increase the expression of these membrane proteins when they need them the most. And we are going to look at enzymes later on in the course, but what you'll usually see is that they will try to function at this lower part of the curve. Okay, so that will really allow them to adapt and accept more substrate so they won't become saturable. That's what we're seeing up here at the top. So whenever a protein is involved, the rate limiting step really is going to be that interaction. You might have heard about channel proteins before, but these are just a few examples transporting things like glycerol or amino acids or chloride ions, and the structure of these membrane proteins is absolutely critical for their function. Some of the key features are that they are incredibly selective. They can select between a potassium and a chloride ion, which if you know the charge you can understand, but they can also select between molecules of the same charge. And when it comes to selecting for one versus the other, it all comes down to the size of the molecule. So these channels have very finely tuned filters that determine what comes in and out of the cell. They can also be incredibly quick and can rapidly move ions at the rate of 100 million ions per second. And I don't even think I can envision 100 million of anything moving that fast. The last thing is that they need to be regulated or gated. And this is very important. You don't necessarily want everything to be open at the same time, or you would just hit equilibrium with things flowing in and out, and there would be no way of maintaining ion gradients. So they can be opened or closed depending on what the cell needs. So the cell does have a way of regulating them and turning them on or off. Rod McKinnon was an MD that actually left med school to do research, and this actually resulted in a Nobel Prize. So I know many of you might want to go into medicine, but maybe take this time to really examine what you're passionate about and what you like to learn about, as well as the types of work environments that you would prefer once we do get back to regular society. He was really interested in understanding how ion channels work, since they're involved in a variety of things like regulating cell volume, the secretion of hormones, as well as the firing of neurons and axons, which is driven by these ion channels opening and closing. When he left medicine for research, there was a general understanding of how things open and close, but he couldn't wrap his head around what was going on at the structural level. Peter Agre also worked on a different channel called the aquaporin channel, which regulates the movement of water, and together they were awarded the 2003 Nobel Prize in Chemistry for helping elucidate what's going on at the molecular level and understanding how these channels can select for different ions and move them across the lipid bilayer. The potassium channel is actually composed of 4 identical subunits, so it's a homotetramer. And these subunits are just shown in different colors, so either in this side view over here, and so that these lines represent the lipid bilayer, or the top-down view. Okay, so when these 4 subunits come together, it kind of forms this cone shape, where the larger opening up here is actually facing the outside of the cell. Okay, so this is where ions would come through. Now, when we look at the top-down view, so if you're looking from the top down here, you can actually see a hole in the middle, okay? And this is essentially what an ion would see if it's looking to get across that membrane. So you can kind of see the relationship of the individual subunits, but it doesn't really tell you how it selects for potassium. So how do we know a potassium ion is going to bind in here versus, say, some other small molecule? Now, what they didn't know at the time is that this is actually the closed conformation. So even though there's a little bit of a hole, it does actually have to open up wider for potassium to get across. And remember how I mentioned that these structures need to be regulated or gated? This is actually what it looks like when it's not transporting potassium. So if you were to look at sort of the surface representation, you actually wouldn't see a hole going straight through. Remember, this is just the backbone of our polypeptide chains. There's actually amino acid side chains that would be blocking and not allowing ions to go across the membrane. Now, the real magic in the structure comes down to really understanding the selectivity filter. Okay, people had actually speculated that there's got to be something inside of that channel that's going to select for one ion versus another. And it turns out that it's not actually a defined structure, say an individual alpha helix or a beta sheet. It's actually found in some loops that are contributed by each of the subunits. So this is just a cutaway view of 2 of the 4 monomers, just for simplicity. But each of the monomers is going to contribute a polypeptide loop that's shown here. And that actually narrows the channel, and it only allows for the green potassium ions to bind. Now, the primary amino acid sequence was known at the time, and people had actually done mutations and made changes to the sequence and found out that it's this sequence here, so the TVGYG, that's going to be important for the activity. So remember, we always write polypeptides from the end to the C terminus. So we've got threonine, valine, glycine, tyrosine, glycine. Okay, and so this is in the middle of the chain, just on this loop here. Now, it was a bit confusing because you couldn't really understand how something like glycine, which just has that hydrogen side chain, could be important for binding to these charged ions. You would maybe expect that there might be some negatively charged amino acids that play a role in this filtration, okay, or only allowing potassium to cross. Now, it turns out that it's not the side chains, but actually the backbone carbonyls. So the C double bond O, so oxygen is just shown in pink here. And these are pointing in alongside with threonine side chain hydroxyl, which is actually at the bottom of the filter down here. And these are going to be playing a role in that selectivity filter, so I hope you can really appreciate just how novel this finding was. Okay, nobody really expected it to be the backbone of the polypeptide that was playing a role. Okay, and it comes down to really the geometry and the chemistry at play, along, which really allows these functional groups to interact with the potassium ion. Now, the other thing to keep in mind is that the potassium ion actually becomes dehydrated. Okay, so normally when it's in solution, because it's charged, it's going to be interacting with water. It's very soluble in water. For it to actually be transported, it needs to be released from that water. So it's got a shell of water around it. When it's floating around a solution, it's going to release those water molecules so that it can fit in the middle of this pore. So if you were to look at the geometry of binding, the carbonyls are actually replacing the bound water molecules as potassium enters into the channel. And there's still actually going to be a water molecule between the potassium ions that's going to be transported. Remember, this is in 3D. But the waters on the sides over here are going to get replaced by those carbonyls. Now, this also makes sense because there would be some electrostatic repulsion between these 2 potassium ions. So there is actually water in between them that is flowing through the potassium channel across the membrane. Now, if you change the sequence of this selectivity filter, or the reentrance loop, which is another name for that selectivity filter, you can actually change the ion selectivity of this cation channel. So here I'm just showing you 3 different sequences, and you can still see some of those conserved glycine residues in pink, as well as some of the other amino acid changes that allow for different ions to pass through the channel. So changes to the side chains that don't directly interact with potassium can actually affect the overall structure of this peptide loop. Okay, and it's going to be the size of these side chains that are affecting the orientation of those backbone carbonyls. So they're still contributing to the structure of the reentrant loop, but not necessarily having a direct interaction with the potassium ion. Okay, and the only exception to that would be that threonine side chain, which can also be mutated to a leucine, and that results in the movement of calcium across the membrane. So this stretch of 5 amino acids here is really forming the scaffold for transport and acts as that selectivity filter. The slide's just showing you the major conformational changes that occur between the closed state that we just looked at and the open state of the potassium channel. So years later, Rod McKinnon actually did publish the structure of the open state. It's not as nice, but what we see is that the selectivity filter is actually still intact when it's in the open state. So it doesn't really collapse, okay? These red loops don't move significantly. They're still able to ensure that only potassium is transported across the membrane. Now, what does happen is that the base is actually now wide open, and those ions are free to travel across the membrane and into the cell. There's also a key glycine residue, glycine 99, remember that small amino acid, that's found in one of the two helices, and this is actually what's allowing the bottom part of the helix to hinge or move out of the way. So we get this movement to allow ions to cross. Now, in the case of the potassium channel, the gating stimulus is actually a change in voltage or pH. So there's a sensor in the channel, so an amino acid side chain, that can actually sense these changes to allow for these conformational changes to occur in the helices and then open the channel and allow for potassium to cross the membrane. Okay, so it's really this helix bending and rearrangement deep in the membrane, because glycine 99 is found in sort of this longer helix that's going to open the potassium channel for transport. We've talked a lot about the structure of membrane proteins, and for the most part they've been alpha helical, but not all membrane proteins have this structure. You can also see beta barrels that consist of many beta strands, and remember, this is the type of domain that'll form a pore that allows molecules to cross the membrane. These membrane proteins are a little different in that they don't undergo major structural changes. When they're present, for instance, porins that are found in the outer membrane of bacteria, they are less selective and can let hydrophilic molecules like glucose into the cell. So they really aren't as stringent as the potassium channel that we just looked at. An important point is that there are similar patterns in their amino acid sequence. So remember that beta strands have a more elongated structure, and for a beta barrel to form, you would have an alternating pattern of one hydrophobic and one hydrophilic amino acid, so that every other side chain would be hydrophilic and pointing into the aqueous middle of the barrel. So if you see a sequence of alternating hydrophobic and hydrophilic amino acids in the primary sequence of a membrane protein, you could maybe predict that it would be found in a beta barrel. Active transport involves the movement of molecules against their concentration gradient. So in these images, you can just see some membrane proteins that need either the input of energy, okay, so from ATP or say light, or maybe even the passing of electrons, in order to undergo these conformational changes to move an ion against its concentration gradient. Now, these are sometimes referred to as pumps, and you can see that they do interconvert between two conformational states. Now, we've already seen a few examples of primary active transport in the course, but it'll also come up in the metabolism section when we look at the electron transport chain. Secondary active transport basically just uses the concentration gradient of another molecule to move one across the membrane. So you're actually going to have two different binding sites for the co-transport of two molecules. So for instance, A could be going down its concentration gradient in one direction, which results in a conformational change in the membrane protein, allowing for B to go up its concentration gradient. Okay, and so this transport can either be in opposite directions, or it could be in the same direction for a symporter. Okay, so an antiporter would be in opposite directions, and a symporter would be moving both molecules in the same direction, using the concentration of one molecule's concentration gradient to move the other one up its concentration gradient. There are two different ways ATP can help mediate this change in a membrane protein. So when ATP is actually hydrolyzed, the free energy that gets released is actually going to be used to change the structure of the membrane protein. Okay, and that's what's really going to allow for the movement of the molecule across a membrane. Now, we saw an example where CFTR can also bind to ATP, and this part of the protein was known as the nucleotide binding domain, or NBD, where the hydrolysis of ATP to ADP and inorganic phosphate was allowing for this conformational change to take place, and that was allowing for the transport of chloride ions. Okay? And you'll sometimes hear these types of membrane proteins referred to as ABC transporters. The multi-drug resistance proteins are also going to use this mechanism to export drugs out of the cell. Now, the other way that ATP can drive the active transport is by donating a phosphate, and that phosphorylated amino acid side chain is going to change the conformation of the entire molecule and allow for the substance or the ion to be transported across the cell. Okay, and so these membrane proteins are known as P-type ATPases. So it really depends on whether or not that phosphate is going to get released, or whether or not it binds to an amino acid side chain and helps the protein undergo a conformational change. Here's just an example of a flipase where ATP hydrolysis is helping to change the conformation of the structure, allowing for the movement of a phospholipid from one side of the membrane to the other. On the left is the open form where ATP isn't bound, and then once ATP does bind, this is what's helping drive or change that conformation in the membrane protein structure, allowing for the lipids to be transported from one face to the other. The last example I want to discuss is bacterial rhodopsin, which we already saw had 7 transmembrane helices. Now, it also has a retinol cofactor that not only contributes to its purple color, okay, but it can also undergo a conformational change in the presence of light. Okay? And so this is really an example of a light-induced transporter that actually helps with the transport of protons across a membrane. And in this case, it's going up the concentration gradient. Bacteria are going to use this proton gradient to then generate ATP. So those protons will then come back down through ATP synthase, and this would be an example of facilitated diffusion where another conformational change would be used in order to generate ATP. When we were looking at hydropathy plots, remember bacterial rhodopsin was one of the examples that has actually 7 transmembrane helices, so that's just what these cylinders are representing, and they're just represented by different letters, so A through G. And there's actually amino acid side chains that are found in these helices that can bind to protons, okay, and that's what's actually going to allow for their transport across the membrane. Now, lysine 216 is actually going to be covalently attached to that retinol cofactor, okay? And when it absorbs light, you can see that this chromophore has lots of double bonds, and you'll actually get a change in its structure that's going to affect nearby amino acid side chains and result in their protonation or deprotonation. So remember when we talked a bit about pKa values and how they can differ depending on what the environment is? This is actually what's going to allow these different functional groups to be protonated or deprotonated and allow for those protons to go across the membrane. Now, this figure is only showing a few of these acidic and basic amino acids, so that lysine over here. But there are other amino acid side chains that are going to help with this proton pumping. What's really neat about looking at bacterial rhodopsin structure and the mechanism of action is that the protons don't actually diffuse across the membrane. They're really hopping or being passed from one side chain to another. Okay, so this is similar to what you would see for free water. So this image is just showing you proton hopping in water alone. So as one hydronium ion gives up its proton, the adjacent one's going to bind to it and accept it. Okay, and then that hydronium ion could then pass one of its other H's onto another water molecule as well. Okay, so this means that true diffusion of the proton across the pump would actually mean that the proton that enters the membrane protein is the same one that's exported immediately across on the other side if you were to track it, kind of like what we saw for the potassium channel. But what bacteriorhodopsin does instead is it uses this hopping mechanism to pass a chain of protons through different amino acid side chains. Okay, so as soon as one proton is picked up from one side of the membrane, another one's going to get released, but it's a different proton that was bound to a different amino acid side chain. That initial proton will eventually exit the transporter, but it's going to be through a series of hops along the different functional groups that allow the transport to occur quite rapidly. So the structure of membrane proteins really is important for understanding their function. We see that conformational changes are key for the movement of ions and molecules across the membrane and can also help with the regulation of transport. Some membrane proteins, like the beta-barrel proteins, can allow multiple molecules to cross the membrane, while other membrane protein transporters are more specific. Transport can either be passive or active, and active transport uses energy or concentration gradients to move molecules across the lipid bilayer up their concentration gradient. Keep in mind that transport is also saturable, so this is all due to the interaction of the molecules with the amino acid side chains found in the proteins. Check out the 2 brain break questions before moving on to the last lecture for the week.